Good evening, everyone. This is fourth semester topic gene therapy for fourth semester BSc microbiology students. So, gene therapy is the introduction of the genes, corrective genes, into the existing cell to prevent the disease or cure the disease. So, gene is going to be defective for a particular protein or an important protein. For example, there are so many genetic disorders which exist. Example is sickle cell anemia. See, it is one of the genetic disorder where the hemoglobin gene becomes defective. So when the hemoglobin gene becomes defective, the right amount of oxygen is not circulated into all the cells and all the tissues in the body. Right, so it causes deprivation of oxygen, breathing problems and all that. So sickle cell anemia is one such genetic disorder. Thalassemia is the other genetic disorder where there are two chains of hemoglobin. One chain of hemoglobin, the gene for one chain of hemoglobin becomes defective. Again, the oxygen circulation becomes difficult in persons. Hemophilia is another genetic disorder where there can be a defective gene for the clotting factor. So the clotting factor is a protein. You know that each protein is coded by a gene. So when there is a defective gene for the clotting factor, the clotting will not happen normally. So that's one of the genetic disorder. The other genetic disorder is severe combined immunodeficiency, where the T cells and B cells are not formed normally. So the person's resistance or immune mechanism is not good. He has defective immune cells in his body. So that's the other genetic disorder. So muscular dystrophy is the other genetic disorder where the dystrophin protein, the gene for the dystrophin protein may become defective. He has fatigue or weakness in any of the muscles in the body. So that is muscular dystrophy. All these are some of the examples. Cystic fibrosis is another important genetic disorder where the persons cannot breathe normally because of the defective protein in the lungs. So all these are some of the genetic disorders. So gene therapy is one of the technique to correct these defective genes by introducing the corrective genes into the existing cell. So there are so many approaches for gene therapy. So there are two approaches like somatic cell line therapy and germline gene therapy. So in somatic cell line therapy, where the corrective genes are introduced into the somatic cells like bone marrow cells or blood cells or skin cells. Right, and in germline therapy, where the corrective gene is introduced into the germ cells like the eggs and the sperms. So in case of somatic cell line therapy, it's not going to be hereditary. It will not be passed on to the next generation. But if the gene therapy is done in the germline cells, then it will be passed on to the future generations also. That's the difference between the two. But currently, they're doing research, but they're doing it for somatic cells, and for genetic cells it's not yet been done, or the research is underway due to many ethical reasons. Yeah, and there are other approaches like ex vivo gene therapy and in vivo gene therapy. So what is done in the ex vivo gene therapy? So the bone marrow cells or liver cells are taken out from the patient's body, and the corrective gene is going to be introduced into these cultured cells. Bone marrow stem cells can be removed from the body, and the corrective gene is introduced into the stem cells, right? And these stem cells are grown in the laboratory, and the corrective gene can be introduced through the virus. Then it is called a viral vector, or it can also be done through the non-viral vector method. So when you do it through the viral vector method, that virus, particular virus like retrovirus, if you use, it has to be modified in such a way that it's not going to cause the severe infection and the disease, but it's going to transfer only the corrective gene into the cultured cells. And once the corrective genes are introduced through the virus vector into the grown cells, then these cells can be reimplanted into the patient's body, and the corrective cells are going to produce the corrective gene, and the genetic disorder can be corrected. So this is one approach. And there is another approach like in vivo gene therapy. So when you do in vivo gene therapy, see, for example, this can be done for such cells where the cells cannot be removed, and even if you grow them in the laboratory, they're not grown normally. right? And even if you introduce the corrective gene into these cells which you have taken out, they cannot be reimplanted into the body without the defects. In such cases, you will go for in vivo gene therapy. So in case of in vivo gene therapy, what do? So again, you can use the viral vector or non-viral vector mechanism, and you can introduce the corrective gene into the defective cells directly into the body, and the corrective cells are going to produce the correct protein. So this is nothing but the in vivo gene therapy. And what are the other, to introduce the genes into the cells, there are many methods like gene gun method. In gene gun method, where the gold or the tungsten particles are coated with the DNA, and the genes are going to be transferred in a high pressure vacuum system. And then the other method is a micro injection method, where you are going to transfer or deliver the DNA through micropipettes, glass micropipettes, and this can be monitored very carefully by observing through the microscope. So this is nothing but micro injection method, right? And so these are all some of the methods where you can use, other than the vectors, non-viral vector methods, to transfer the DNA or the corrective genes into the systems, right? And there are many advantages of gene therapy. So the defective gene can be corrected, it can be long lasting, and the genetic disorders can be corrected in the prenatal diagnosis itself. All these are some of the very good advantages, so the person can lead a normal life. But what may be the disadvantages of the gene therapy? The disadvantages may be, see, when you use a virus, you don't know that may start the replication. If you are using a pathogenic virus, that may start the normal cycle and normal replication process in the person's body or in the patient's body, and he may not be immune to that particular virus, and the disease may be developed in that person, right? And he may suffer from the disease which is caused by the virus. And when you do it for germ cells, you do not know the effects, right, after so many years. So the effects can be anything, okay? It's non-predictable in such cases when you do for germ cells. So these are all some of the disadvantages. So you can use many methods to transfer the genes, either through the viral vector system or through the non-viral vector system, right? And what are the other methods for gene therapy? So gene therapy, you can do it with somatics. Gene augmentation therapy is there, where a defective gene can be augmented or it can be corrected by transferring the corrective gene. So the defective gene is not going to be removed, but the effect of the defective gene can be deleted or it can be corrected by transferring the corrective gene into the cells where a defective gene can be corrected. So this is done in case of the gene augmentation therapy. That can be one of the method. And there can be other methods like gene inhibition therapy, where you are going to inhibit the activity of the defective gene. See, this can be done by anti-gene therapy method. So in case of anti-gene therapy method, so the transcription can be corrected by transferring the antigene, okay, by transferring the oligonucleotide for the antigene. So this is nothing but antigene therapy. So the other method is nothing but antisense therapy, where during the protein synthesis, the translation process itself can be corrected by sending the antisense gene. So by giving the antisense gene, so this antisense therapy can also be used for the gene therapy. So there are n number of genetic disorders are detected in so many persons, and the persons suffer from severe immunodeficiency and other diseases. This has even been tried for some of the oncogenic genes, so that the trials are being underway and the research is going on even to detect the oncogenic genes in case of the persons, or in the prenatal diagnosis of the oncogenes can also be detected. So all this becomes possible. So this may give way for many other important applications right? So the defective genes can be corrected and the person can lead a normal life due to this one of the important technique called gene therapy, where a defective gene is corrected. So that's about the gene therapy.